The study is very important because it was the first large clinical study showing how this new test functions in actual daily practice. And the investigators at USMD, which is a large physicians group in Dallas, Texas, in the United States, selected 400 patients of Medicare age, 65 years of age and older, who were resistant to having been screened for colon cancer. They were what we call non-compliant. They hadn't had a colonoscopy within 10 years or stool blood testing. And we applied the test to those patients, and 88% of them did the test. Importantly, 90% of them who were positive on the test complied and went through and got a follow-up colonoscopy, which is what you want them to get. 4 of them had cancer, all early stage, all surgically curable. 21 had the type of lesions we call advanced polyps, advanced adenomas, that are more likely to become cancer in the lifetime of the patient. Important disease. Oh, yes, yes. So what was most interesting about this test was used in actual daily practice. The test itself was approved by the United States Food and Drug Administration, the FDA, in 2014. It's covered by our Medicare, our government insurance for patients 65 years of age and older, without copay or deductible, as a screening option that they can enjoy. So it's in use now. The observation that there are DNA fragments in the stool stream that originate in the colon, specifically originate in polyps, premalignant lesions, and cancers, has been described since the early 90s by many folks. Our key collaborators were key in that, the Bert Vogelstein lab at Johns Hopkins and many others. And working with that, we've been working with it now for about 17 or 18 years. In order to devise the mechanics of using a stool DNA analysis that tells us that these lesions may be present. So the DNA that we're identifying, and human DNA is very rare in stool. There's not much there. We actually reabsorb most of it and recycle it. It's critical because it's coming from the lesions. There's a little bit of background abnormal DNA that comes from the colonic lining, but for the most part, this is somatic mutations and alterations, epigenetic changes that happen as part of the developmental process of the cancer. So it's biologically related to carcinogenesis, as opposed to nonspecific findings like blood, which are related to ulceration as lesions get larger. It wasn't easy because there were a number of analytical challenges that had to be accounted for. Number one, these are very rare molecules. The ratio of human DNA to bacterial DNA is about 1 part to 100,000. Okay, so we had to isolate the DNA in a way that we could amplify it so that we could interrogate it for methylation changes and for mutations. Once we did that, there were several other issues that had to be addressed with how you run a methylation assay. Could you multiplex the assay? Could you make it reproducible? Could you package it and make it so that any lab could do it day after day after day? In clinical pathology, that's what we do. We have things that are very robust. We addressed all of those things, and that's just on the analytics side. Patient preference and patient utilization is key. You can have the best test in the world, and if no one uses it, it's not very effective. So we talked to literally hundreds to thousands of patients. As we said, well, if you're going to collect a specimen, how should that be? So we've eliminated everything that was a problem to the extent possible. There's no preparation. There's no change in medication. There's no change in diet. It's random access. You pass a portion of stool into a container that's been purposely designed to make it easy for patients. They deal with the sample. It goes back in the box it came in and gets shipped off back to the lab, all prepaid. Privacy of your own home, no schedule, no time off from work. There's a lot of people, and whenever you have a new test, it still takes time to get in contact with hundreds of thousands of primary care physicians in the United States alone to advise them of the test, how the test works. They have to have, reimbursement, et cetera, et cetera. So I would say that things are going along, as you might say, swimmingly. They're going along quite well as we've rolled the test out. Probably 35 to 40,000 physicians now have ordered the test over time. we've done in the well over 100,000 of these tests just in the first year of its availability. So things are going well, but most importantly, about 60% of the patients who are using this test so far, age 50 to 65, have never elected to be screened before. Age 50 to 75, the sweet spot for screening, 42%. So importantly, the test is already making a difference in bringing patients into the screen pool who've never been screened before. Very gratifying. When we meet again in 5 years, hopefully we'll be able to say that 90% of the patients in the United States of screening age have been effectively screened.